Alright, who's ready to learn some biochem? So this is going to be your final exam. It's going to cover chapter 15, 16, 17, and 18. So chapter 15 is translation. So we're going to start with learning objective 1. So this is the picture is very important to understand. Describe the universal flow of genetic information. So we know the universal flow by just looking at the picture is DNA to RNA and then to protein. So starting at the beginning, DNA is your genetic information, and we get that by DNA replication. Once we have DNA, we go through transcription to get RNA. So DNA transcribe to RNA, and RNA is your transmitter of genetic information. So look at the circle around it. You see that replication and transcription both occur inside the nucleus. So once you're outside in the cytoplasm, you go from RNA to protein, and that's called translation, and that's our main focus for this chapter. And then protein is your biocatalyst. And then also for learning objective 1, you need to know the exception to this rule, and that is RNA going back to DNA. So that's called reverse transcription, and you've seen that before in viruses. Alright, so here are your learning objectives. We already went through one, so let's get started. Alright, so learning objective 2 is to know why the genetic code is degenerate, unambiguous, non-overlapping, and almost always universal. So that statement is very important to remember for the exam. He might switch it up and say ambiguous instead of unambiguous, or he might say always universal instead of almost always universal. So just pay attention to that statement and understand why the genetic code is that. So it's degenerate because each amino acid may have more than one codon. So for example, phenylalanine, it has two codons. It has UUU and it has UUC. So that's what makes it degenerate. So why is it ambiguous? Each codon specifies only for one amino acid, so that makes it unambiguous. So if you look at UUU, that codon specifies only for phenylalanine. If you look at AUG, that codon only specifies for methionine. Okay, so the next one is that it is non-overlapping. So mRNA doesn't contain any extra nucleotides. It's read sequentially starting from the start codon, AUG, and then ending at the stop codons, UAA, UAG, and UGA. So none of them will ever overlap. And then it's almost always universal because most of the times the codons will specify for a certain amino acid, but in some cases, like in human mitochondrial mRNA, UGA, which is normally a stop codon, actually specifies for tryptophan. So that's what makes it almost always universal. And then on the very bottom right corner, it's important to know that methionine and tryptophan only have one codon, whereas the other 18 amino acids have multiple codons. So the key thing to take away from this slide is to know that genetic code, it's degenerate, unambiguous, non-overlapping, almost always universal. Know your start codon, which is AUG. Know your three stop codons, which is UAA, UAG, and UGA, and then know that methionine and tryptophan only have one codon. So this slide's just talking about something called wobbling, which affects the third base of the codon. So base pairs can wobble. For example, G can pair with U, and AC or U can pair with a base called hypoxanthine, which is represented by I, and it's found in tRNA. So for example, here we have alanine. We know alanine has 4 different codons, if you look back at the previous table. And it's saying that 3 of the 4 codons, GCU, GCC, and GCA, can pair with a single tRNA that contains the anticodon IGC. And that's shown at the picture on the bottom. So if each of the 61 codons for amino acids require a distinct tRNA, then the cells would contain 61 tRNAs. But because of wobble between the codon and anticodon, fewer than 61 tRNAs are required to translate the genetic code. So this isn't a learning objective, but it just talks about the reading frame of mRNA. So if you look at picture A, you have 3 different reading frames for any given mRNA sequence. If you look at picture B, that's the one that we need to start translation. So AUG, remember that is the start codon, so it sets the reading frame for translation, and then the other reading frames are not used at all. So here's learning objective 2. Describe the interaction of codons and anticodons and the relationship to mRNA and tRNA. So in the picture, at the bottom we have mRNA. Know that mRNA contains your codon. So in this case you see AUG, remember that's your start codon. And then at the top we have a cloverleaf structure called tRNA. And tRNA contains your anticodon. So key things to remember: mRNA contains your codon, tRNA contains your anticodon. And then just remember that pairing is complementary and anti-parallel. So here again, it's just the same concepts. Anticodons on tRNA, and it's anti-parallel and complementary to the codon, which is translated on mRNA. Wobble, we've talked about that earlier. That takes place on the 3rd position of the codon. And here it's saying that the 3rd position of the codon does not always need to be paired with the anticodon. So here we have learning objective 4, knowing the different types of mutations in RNA and DNA and knowing their related diseases. So first we have a point mutation, which means a single base will change. So there's actually 3 basic types of point mutation, and they are silent, missense, and nonsense mutations. So all 3 of those are types of point mutations. So silent mutation is just a change that specifies the same amino acid. So if you look at the example, CGA changes to CGG, you still have the same amino acid, so the amino acid sequence isn't affected. So that's a silent mutation. The next one is missense mutation, and a change that specifies a different amino acid. So a codon change from CGA to CCA will change arginine to proline. So that's a missense mutation. And then you have a nonsense mutation, and that's a change that produces a stop codon. So if the codon CGA changes to UGA, that's going to make your amino acid arginine change into a stop codon. So those are the 3 types of point mutations. And then you also have insertion, which is addition of 1 or more bases, and then you have deletion, which is a loss of 1 or more bases. So this is a frame shift mutation. Basically, it can be an insertion or it can be a deletion. In this case, it's a base insertion. So you see A is inserted into the reading frame, which causes a shift to the reading frame. So anything after where that A is inserted in can have an effect on the protein that's being translated. So here's a good slide to memorize. This is still part of learning objective 4, knowing the diseases that can be caused by these mutations. So silent mutations, we know that it's not going to be affected since the new codon is going to specify for the same amino acid, so no problem there. Missense mutation, however, remember the new codon is going to specify for a different amino acid, and in this case it's going to cause sickle cell anemia. So you should know that a missense mutation could lead into sickle cell anemia. Nonsense mutation, when you get to the stop codon, becomes non-functional. Frame shift, deletion or addition, also non-functional. Then you have large segment deletions. So if you delete a large segment of your amino acid sequence, that could cause loss of function, or for example the disease alpha thalassemia. Mutations in your splice sites can cause beta thalassemia, Tay-Sachs disease, Gaucher's disease, and then triplet repeat expansion is going to lead to Huntington disease, spinal bulbar muscular atrophy, Fragile X syndrome, myotonic dystrophy. So pay attention to which diseases are going to be caused by the different types of mutations. Especially for the triplet repeat expansion, pay attention to which ones are for the coding region and then which ones are for the non-coding region. So here's another learning objective, knowing the formation of aminoacyl-tRNA. So tRNA has to be charged before it can bind to the mRNA. So whenever it's charged, it's called aminoacyl-tRNA. So how does it become charged? We need to add an amino acid. So all we're doing here is adding an amino acid to the tRNA. So how do we do that? The first bullet says each type of amino acid is activated by different aminoacyl-tRNA synthetase. So that aminoacyl-tRNA synthetase is going to be specific for each amino acid. So there's 20 amino acids. That means that there's going to be 20 different aminoacyl-tRNA synthetases. So that synthetase is going to take your amino acid and it's going to transfer it to the 3' end of your tRNA. So that's what the second bullet says. Aminoacyl-tRNA synthetase transfers the activated amino acid to the 3' end. And then it's going to form an ester bond, which is going to link it to the tRNA. And that's how you get aminoacyl-tRNA. So to recap, you have a tRNA and you have an amino acid. How do we get that amino acid bound to the tRNA? We need a aminoacyl-tRNA synthetase, which is specific for each amino acid, and it's going to transfer the amino acid to the 3' end of your tRNA by the formation of an ester bond. So that's how we form a activated aminoacyl-tRNA. And the third bullet is just saying that the tRNA synthetase has self-checking functions. So if it's the wrong amino acid, then it's going to kick it off and find the correct amino acid. Because remember, there's 20 amino acids, so there's going to be 20 different aminoacyl-tRNA synthetases. So here's just a picture of the tRNA. If you pay closer attention, there are highlighted green areas, and those are called recognition sites. So in the previous slide, we talked about making the aminoacyl-tRNA. But how does the synthetase know where to put the amino acid? So it knows where to put it because there are recognition sites on the aminoacyl-tRNA. And also, if you read in the notes below, in some cases, the anticodon can act as the recognition site. So this is just a 3D structure of tRNA. You don't need to pay attention to this. So this is Learning Objective 5, describing the 3 phases of translation. So we have initiation, elongation, and termination. So pay attention to this slide, it's very important. No keywords. So initiation is often called the rate-limiting step. So this needs to occur first, otherwise elongation and termination can't occur at all. So initiation also involves the small and large ribosomal units. Second step is elongation. So that's where the protein synthesis from the N to C terminal begins, and this is where the ribosome translocates along mRNA. The last phase of translation is called termination, and remember those 3 stop codons I told you to memorize? They're going to come back, so terminates at those 3 sites, UAA, UAG, and UGA, and that's where it's going to release the large and small ribosomal subunits that found earlier in initiation. Table, but I don't recall any of this being on the test. If he said anything in class about this, then maybe focus on it, but I don't see this coming into the test. So here's an overview of translation. I highly recommend watching a video on translation so that you can visualize things better. So just take a few minutes and go do that. Here, first step is a tRNA donated its amino acid and is now being released from the mRNA. So in step 2, another tRNA, remember the charged tRNA called the aminoacyl-tRNA, is going to bind to the correct codon and donate its amino acid to the growing polypeptide chain. So we started with methionine, then valine, then proline, then glutamine, and now aspartate is joining the chain. So here's a simplified version of the initiation of protein synthesis. Sorry if the slide is blurry, just follow along on your own notes. And this isn't a learning objective, but it's good to know. So follow along. We'll start off with methynyl-tRNA. So that's that yellow macaroni structure at the very bottom left. So methynyl-tRNA, remember methionine is the amino acid for the codon AUG, which is your start codon. So that's why we start with methionyl-tRNA. So methionyl-tRNA binds to EIF2, which stands for Eukaryotic Initiation Factor 2. So those two become a complex, and it's going to bind to EIF3, which is that red circle that's bound to the small ribosomal subunit, which is the 40S subunit. So quick recap, methionyl-tRNA binds with EIF2. That complex is going to bind to EIF3, which is bound to the 40S ribosomal subunit. Okay, so now we have this complex that has EIF3, the 40S ribosomal subunit, methionyl-tRNA, and EIF2. Okay, so that's one complex. On the other side, on your mRNA, we have the 5' cap, and it's going to form this cap binding complex with EIF4, and it's going to do that by hydrolysis. So once that 5' cap binds with the EIF4 through hydrolysis, it's going to combine with that other complex we made earlier. So once all of those combine together, it's going to call in the 60S subunit. So once the 60S comes in, it's going to become a complete ribosome, and now you have 3 binding sites for the tRNA to come in. So you got the P site, which is the peptidyl, you have the A site, the aminoacyl, and then the E site, which is ejection. And we're going to talk about those soon. So this is the step after initiation. This is elongation. I'll touch base on the important concepts, but you don't have to know everything in super detail. So we know met-tRNA is already in the P site. So the mRNA codon in the A site is going to determine which aminoacyl-tRNA binds to that A site. So in the example, we have the codon GUU. So that's going to code for valine. So we're going to need a valyl tRNA to bind to the A site. But before it can bind to the A site, it needs to form a complex, and it forms a complex with elongation factor 1A. And then it also needs GTP. So once all that's done, it's going to bind to the A site, and in step 2, it's going to form the first peptide bond. So pay attention to this. This is going to be a test question. Where does the formation of the first peptide bond occur? So if you look at the picture, you can see that peptide bond forms in the A site. Okay? So in step 3, everything's going to translocate. So whatever was in the P site is now in the E site. Whatever's in the A site is now in the P site. So I think this is an error because it says subsequent formation of a peptide bond occurs in the A site. It should be in the P site. So just double check with Dr. Schroeder, but even the picture shows that subsequent peptide bonds will form in the P site. And it even says that on the next slide. So the first peptide bond forms in the A site, but subsequent formation of the peptide bond forms in the P site. So just double check that. I'm pretty sure, but not 100%. And then after step 3, everything is going to repeat itself. So formation of that peptide bond that we saw earlier. Remember, the first peptide bond is formed in the A site. If you look at the third bullet, it says in subsequent rounds of elongation, the amino acid on the tRNA in the A site now forms that peptide bond on the tRNA in the P site. So I'm pretty sure he made a mistake. Make sure to double check that. So what forms that peptide bond is something called a peptidyl transferase. So that's an enzyme that is going to catalyze the formation of the peptide bond. So the uncharged tRNA moves from the P site to the E site, so that happens during translocation, and it's going to be released from the ribosome so that the next charged tRNA can enter the A site. And then this is going to repeat until you have a nice and long protein chain. So earlier we talked about the elongation factor 1, how it has to bind to the amino acid before it can go into the A site. So our previous example was valyl-tRNA. Remember, before it can bind to the A site, it has to be bound to EEF1A, which also has GTP activity. So once that complex is formed, it will bind into the A site, and your EEF1A is going to leave after GTP is hydrolyzed. So you're going to get EEF1A and GDP, and that in turn is going to bind to EEF1B, which is a guanine nucleotide exchange factor. So it's going to take the GDP that our EEF1A has and exchange it for GTP. So now you have EEF1A and GTP all over again, so that you can continually bind to amino acids so that this cycle can continue. So the last step is termination of translation. So remember the 3 stop codons? So once a stop codon moves into that A site, it's going to tell the peptidyl-transferase to hydrolyze the completed protein. So this slide is just kind of thrown in here. It's not a learning objective, so I wouldn't put too much focus on this for this chapter. So just kind of read through the bullets and then move on. Learning objectives list the post-translational modifications that occur on select amino acids. So I'm not going to read these to you. Just be sure to look at all of them. They could all be fair game for the test, especially the one highlighted in red. So methylation, the addition of a methyl group on the lysine to alter the charge. So that's a type of post-translational modification that occurs on lysine. So you don't need to know this picture for the exam. You can move on. So this slide just talks about the post-translational protein modifications on the side chains. The learning objective is knowing the modifications for the amino acids. So I wouldn't pay attention to this slide unless he said so, but for our exam, he didn't mention any of these. Here's another important slide to pay attention to. This is one of your learning objectives, to know the differences between eukaryotic and prokaryotic protein synthesis. So remember, eukaryotes, they have that 5' cap that binds to the eukaryotic initiation factors. Remember that the first amino acid is methionine, so Aminoacyl-tRNA. Remember, we used the EIFs, and then we have the ribosomes 40S and 60S, which form the 80S ribosomal subunit. Prokaryotes, they have the Shine-Dalgarno sequence. They have formal methionine as their first amino acid. They use initiation factors, and then they have the 70S ribosome. So just pay attention to this slide. Know the key differences, especially those highlighted in red. So this slide wasn't tested on. It's just showing that in prokaryotes, they contain formal methionine as their first amino acid. So here's another key difference. In prokaryotes, transcription and translation can occur simultaneously, but in eukaryotes, transcription has to happen first and then translation follows. So this is another table, but unless he specified in class to know it, this isn't something I would pay attention to. I don't remember needing to know any of this for the test. So yep, this is just another complex structure that you can skip. More pictures. So nothing to know here, just more pictures. This is a picture of the EP and A site of bacterial ribosomes. You can skip this too. Fun fact, pyromycin is an antibiotic that is a toxin. It's another picture, so most of these pictures you can skip. 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 So this is just a model of the eukaryotic ribosome, saying that it is 40% larger than its bacterial counterpart. So remember what makes the 80S eukaryotic ribosome, the 40S, and the 60S. So you've seen this slide before. The main thing is to know what makes the ribosomal subunits. So if I ask what makes the 80S ribosomal subunit, you should know it's the 40S and the 60S. If I ask what composes the 60S, you should know it's the 5.8, the 5, and the 28S, and so on. So you should be able to write all of these down and be able to tell me what makes what. And then also tell me, are they in the nucleolus, are they in the nucleus, or are they in the cytoplasm? So these are the key steps in eukaryotic ribosome synthesis. The next slide explains this a little better. Be sure to read the notes and try and match those up with the picture. These are the key steps in ribosomal synthesis. The easiest way is to look at these bullets and compare it to the picture and just try to follow along. So you have pre-rRNA, that's transcribed from rDNA using RNA polymerase 1. After that, you're going to have modifications, then processing, assembly, diffusion, structural rearrangements, late maturation, and then protein synthesis occurs. So just be familiar with these two slides as they are one of your learning objectives. So here, still part of your learning objective, just know which diseases can cause what. So diamond blackfan anemia might cause problems in the 60S subunit assembly. It may also cause problems in the 40S subunit assembly, shown on the very left. Treacher-Collins syndrome might have problems with rRNA modification, polymerase 1 transcription, etc. So be familiar with those. Here are some of the pictures illustrating the previous diseases. Here's a summary of the diseases. It's a good table to know. For our test, we only had to know column 1 and 3, but I'm not sure what he said for your class. So here again are your diseases. Very good to know. Read through these and be familiar with the features of the disease. Nothing here to know. So here's another one of your learning objectives. Describe events of proteins synthesized on polysomes and rough ER. So what is a polysome? So we know that one ribosome moves along the mRNA, and that produces your polypeptide chain. Well, a second ribosome can bind to the free 5' end of the mRNA. So many ribosomes can actually translate a single mRNA. And when you have multiple ribosomes translating the single mRNA, you form a complex known as the polysome. So a single ribosome can cover approximately 80 nucleotides of mRNA. Therefore, ribosomes can be positioned at every 100 nucleotides. So this is part 2 of your learning objective, knowing the synthesis of proteins on the rough ER. So we already know translation occurs in the cytosol, so that's step 1. In step 2, a signal peptide is going to come out of your ribosome, and a signal recognition particle, known as SRP, binds to it. So SRP is going to bind to your signal peptide, and as soon as that happens, your synthesis of protein is going to be inhibited, so translation stops. In step 3, SRP will bind to an SRP receptor found on the rough ER membrane. So this allows your ribosome to dock on the rough ER, and in step 4, SRP is going to be released, and protein synthesis is going to resume. So step 5, a signal peptidase is going to remove your signal peptide, and then step 6, synthesis of the protein continues, and then you're going to get a completed protein which is released into the lumen of the rough ER. So what happens to the proteins that we make on the rough ER? So the left bullets say that protein synthesized on the ER is going to travel in vesicles to the Golgi complex. So these proteins are destined for 3 things: for lysosomes, cell membranes, or secretion from the cell. So test question could be, what are the fates of proteins synthesized on the rough ER? Answer choices could be destined for lysosomes, cell membranes, or secretion from the cell. So know those 3 well. Conversely, proteins that are synthesized on cytosolic ribosomes are going to be transported into the mitochondria, peroxisomes, and nucleus. So remember, if the proteins are made on the rough ER, they're destined for lysosomes, cell membranes, or secretion from the cell. But if they're made on cytosolic ribosomes, then they're going to be released into the cytosol, mitochondria, peroxisomes, and the nucleus. So these are your last 2 learning objectives. First one is describing the events in protein targeting and how defects in phototransferase activity can lead to eye cell disease. So this is still part of your learning objective, understanding eye cell disease. So it goes by a couple names: eye cell disease, inclusion cell disease, mucolipidosis 2. So you should know all those names. And basically, it's a rare condition where your lysosomal enzymes lack a mannose phosphate marker. So this mannose phosphate marker is important to have. Without it, the lysosomal enzymes don't know where to go. So if you have a lysosome, whatever's inside of it has to be degraded. Otherwise, it accumulates within the membranes. So if your lysosomal enzymes lack that mannose phosphate marker, they don't know how to get to the lysosomes. So that's going to result in an accumulation of things inside your lysosome, and it's going to form inclusion bodies. So the second bullet gets a little more specific. So the reason why the lysosomal enzymes don't have that mannose phosphate marker is all because of a mutation in the enzyme N-acetylglucosamine phosphotransferase. So that's an important enzyme to remember. We'll see more of it on the next slide. But this mutation is required in the first step. So if it's mutated, then we're not going to be able to form that mannose phosphate marker. And as a result, this will lead to lysosomal storage disease and death before the age of 8. So here is just a picture showing you how we can get eye cell disease. So earlier we said it's because of the mutated enzyme N-acetylglucosamine phosphotransferase. So that's defective in eye cell disease. And as you can see, without that enzyme, we cannot get the mannose 6-phosphate at the end. So that serves as our marker for where the lysosomal enzymes need to go. So this is your last learning objective, knowing the different antibiotics and where they act. So we have 4 antibiotics, and let's see their effect on prokaryotes. So streptomycin binds to the 30S ribosomal subunit. It's going to prevent formation of that initiation complex, and then it's also going to cause misreading of mRNA. Tetracycline also binds to 30S subunit, but inhibits binding of the aminoacyl-tRNA to the A site. Chloramphenicol binds to the 50S ribosomal subunit and inhibits the peptidyltransferase. And erythromycin binds to the 50S ribosomal subunit and prevents translocation. So make sure you know which ribosomal subunit it binds to, and then know what is the effect of binding to it. And then, fun fact, did you know streptomycin can cause permanent hearing loss? So using streptomycin is mainly to treat tuberculosis or other infections that don't respond well to other antibiotics. So here's just a picture of the 4 antibiotics that we just saw. You don't have to memorize these for the tests. So here's just a picture showing where those antibiotics inhibit protein synthesis. Focus on the main 4 that we talked about: streptomycin, tetracycline, chloramphenicol, and erythromycin. And then just read through those bullets on the left. So these are inhibitors of eukaryotic translation. I'm not sure if you're supposed to know this, because it is not a learning objective, but if he said to pay attention to these in class, then I would know them. But if you're just following the learning objectives, then this is not one of them. So here is our infamous disease table that we've seen so many times. Make sure to read through your disorders and then the details of each. And we're done with Chapter 15.